previous class where we were talking about the glycogen metabolism or control of glycogen metabolism. But here we will be focusing on specifically the mechanism. And this mechanism is very important for the biological system because it is transferring the information, and so you will be learning that how the information is being signaled or transferred or processed in metabolism. So here you will find that signal transduction cascade mediates the sensing and processing of a stimuli. So suppose there is a stimuli, they will be, amplified, they will be sensed, and they will be processed. So these molecular circuits detect, amplify, integrate diverse external signal to generate responses such as changes in enzymatic activity, gene expression, or ion channel activity. That means they can control. So all the control is happening through the signal. So actually what happens that you have a signal, it should bind to a receptor which is present on the surface of the cell. Then as soon as it binds to the receptor, the signal will be amplified. And once it is amplified, it is transduced. Transduced means it will be in another form. So the information can be, converted into chemical form or some other form. That is what is the transduction. And the same thing you find in case of, other mechanism also. And once this is transduced, there will be response. And once response comes, actually what you require is that this response should not continue. This should be controlled. So there will be mechanism where you can stop this signal transduction, and that can happen even by terminating or by, understanding whether the signal is bound to the receptor or not. So this whole mechanism we are going to discuss today. So here there is a signal transduction. It depends on the molecular circuit, and we are just over reviewing it. I'm not taking a single example and explaining it, but yeah, wherever it comes, we will be talking about the specific example. So first thing is that membrane receptor transfers information from environment to the cell interior. So this means that suppose there is a cell and outside there is a, outer surface of the cell. So information which is coming from outside is being transferred inside the cell through the membrane receptors. So the information that signal molecules are present must be transmitted across the cell membrane. And here, remember that it is not necessary that the molecule which is binding to the receptor themselves should enter in the cell. That means the molecule which is binding, that acts as a primary messenger of the signal. And the binding site on the extracellular domain specifically recognizes the signal molecule. Mostly it is referred as a ligand. And the information, whatever is embodied by the presence of a ligand, is often called as a primary messenger. And it must be transduced into other form that can alter the biochemistry of the cell. To explain this, let me tell you, suppose you have a cell. So what happens that hormones, they are being secreted from glands, and these glands could be in any part of the body. So suppose you have a hormone which has been secreted by the gland. Now what happens, it is going to be moving around the body through the blood circulation. So it is present in the blood, it is a free, it can move anywhere. Now hormone is a primary messenger. What it will do is that it will go and bind to those cells which is having receptor for hormone. So that means if hormone is secreted in the whole and is moving throughout the body, it doesn't work in all the part of the body. It works in only particular part of the body. Suppose a hormone has to work in a liver, it will go and bind to the liver. If it has to work in a particular cell, it will go to and bind to the particular cell. So how that happens? It happens through the receptors which is present on the surface of the cell. So hormone is acting as a ligand. It binds to the surface of the cell, which is a receptor, and once it binds, then only the process is being triggered. So now it says that second messenger relay information from receptor ligand complex. So what it means is that in a cell, if just receptor is present, signal will not move on. There will be no information which will be transferred. If there is a ligand alone, it will not transfer. So important the receptor ligand complex is formed. So as soon as, for example, hormone comes, bind with the receptor, a complex is formed, the relay of information starts. So first, second messenger are often free to diffuse to other compartment of the cell, such as the nucleus, where they can influence gene expression and other processes. Now, there are certain second messengers which will stay up to the outside the cell. So it will just bind to the receptor and this is over. On the other hand, there are some hormones which can bind with the receptor, and along with the receptor they enter inside the cell, go to the nucleus, bind, and can change the transcription process. Means they will actually go inside the cell and they have a specific binding place, especially in a nucleus, where they will influence gene expression. So they can express they can also remain outside the cell and influence the enzymatic activity. The signal may be amplified significantly in generation of second messenger. So what it means is that suppose there is one molecule of hormone, it can produce several molecules of second messenger. And I will come to the what are the second messengers. Thus, what happens, the advantage of this process is that by binding itself, suppose hormone comes, bind with the receptor, it will increase several second messengers. It could be cyclic AMP, it could be G protein, it could be, others. So we will talk about that. So what can happen that input from several signaling pathway can often lead to the crosstalk and may affect the concentration of a common second messenger. So suppose this is suppose a signal has to be increased and there are two different messages which are coming, they can lead to the amplification of signal. So it's a very interesting and complicating mechanism which is showing how the control occurs inside the cell. So far we have talked about how the hormone comes bind with the receptor. Now there is a common message or common method by which you can see these second messengers they work. Many times the second messenger elicit a response by activating protein kinases. You have seen this thing in the previous class where phosphorylase B is activated by protein kinase A. The same way, you can have a protein kinase. The reason here is protein kinase could be one, but it can activate many proteins or many enzymes because its job is to phosphorylate. And because it can phosphorylate, it can activate or inactivate. Both can happen. In one case, you have seen it activates. In another case, you have seen that protein kinase can inactivate. So it can control the reaction. So actually, what I'm saying is that it can control the reaction. Now, it is also interesting that because protein kinase just phosphorylates, so phosphorylation, if it activates, it can be deactivated also by another enzyme, which is a phosphatase. So protein phosphatase and protein kinase, they interplay with each other. So one can activate, another can deactivate. And how this phosphatase works, you have seen this phosphatase just removes one phosphoryl group from the protein, and then protein becomes inactivated or activated. Both things are happening by this process. So what happens when it happens? When this kind of a modification occurs, the signal is terminated. For example, if kinase is activating an enzyme, the protein phosphatase, they can deactivate it or terminate the signal. The signaling process that failed to terminate properly is really dangerous because it may lead to uncontrolled cell growth, which can lead to the cancer. Suppose a signal comes from hormone that cells should divide, should increase in the number. But if we do not terminate the signal, what will happen? It will make a tumor. It will make a cancer. So it is very important that termination should also occur. So here you have learned that in signal transduction, both events are important. First is that it should activate the process. It should, Signal should pass on the information, but at the same time, once job is over, signal should be terminated. Now, looking at the different types of second messenger. Suppose a signal is coming from your this hormone, then you should also have the second messenger which can carry the information. For example, these are second messenger. One is a cyclic AMP or cyclic GMP. It is coming from where? The ATP. Once we hydrolyze ATP, it can be, you can form ADP and PPI, or you can remove PPI and you can form cyclic AMP. And if I start from GTP, I can form cyclic GMP. So both are acting as a second messenger. So these are examples of second messenger. Another second messenger is a calcium ion, which can, uh, improve or activate enzyme, and that's the way they can control the reaction. Third one is inositol triphosphate, inositol 145 triphosphate. What is inositol triphosphate? You have learned that there is one of the phospholipid which is having inositol, phosphatidyl inositol. Now, phosphatidyl inositol, if I remove the inositol, you get inositol triphosphate. So this is coming from where we are talking about this inositol triphosphate. This is coming from the membrane. So membrane itself has a phospholipid. One of the phospholipid could be phosphatidyl inositol. So suppose a second messenger, one messenger comes, primary messenger comes, bind with the receptor and removes this IP3, that is inositol triphosphate. This becomes as a second messenger. Same way diacyl glycerol. These inositol triphosphate and diacyl glycerol, they are the product of the same molecule. So suppose I have phosphatidyl inositol, I remove IP3 from there, the rest portion is diacyl glycerol. So these two are coming from the hydrolysis of phospholipid present on the membrane. This is something which shows you molecular circuit and molecular switch. What it gives you very basic information. Excuse me. So here it shows that how molecular switch operates. In most of the cases, it has been learned that it is the hormone signal or a signal coming from outside the cell is mediated by a protein called as a G protein. So what this G refers to? G is the one which allows the binding with a GDP or GTP. So normally, when this G protein is present in inactive state, it is bound to GDP. And it is a trimeric. There is a purple color is one, blue color is the second one, and this yellow color is third one. So these three are subunit of G protein. Now what happens that as soon as hormone binds with the receptor, I will show in the next slide, hormone receptor binding, what it does is that it triggers the change in G protein in such a way that these two parts, blue and yellow part, is removed. And at the same time, GDP is exchanged by GTP. So instead of GDP binding to this protein, GTP binds. So this is the exchange reaction which occurs. And then what will happen? GTP will bind, GDP will be removed. And this binding of GTP releases these two subunits. So suppose I say that this is alpha subunit, this is a beta subunit, this is a gamma subunit. So what will happen that it will release the alpha subunit, and beta and gamma subunit will remain intact. Once this dissociation of G protein occurs, the GTP bound alpha subunit becomes active. Now this active protein will activate a talk in the next slide. But what I'm showing here is that the same G protein can exist in off position and on position. In off position, all the trimer is present, all the subunits are present. In on position, two subunits have been released. Now, if it has to recover its own function, it has to be your alpha and beta subunit should bind. So in that case, the hydrolysis of GTP Slowly, this hydrolysis occurs because the protein has enzymatic activity which can hydrolyze GTP. And once it is hydrolyzed, what will happen? It will become GDP, and the two subunit which was released here, they will come and bind. So they will come back to the off position. So this way, the triggering of off to on position occurs, and this happens because of the binding of the hormone to the receptor. So what are these receptors? These receptors are 7 transmembrane helix. I'll just show it to you. These are 7 transmembrane helix. So these are present across the membrane. These are integral protein present in the membrane. So these are the one where the binding occurs. So so this is the one which is shown in the detail. And what happens to this? This is the example where rhodopsin is working. Rhodopsin is a protein which is three-dimensional structure. Rhodopsin has been shown, which binds with the 7-TM receptor, and binding increases the, catalyzes all the events. So it is the one which is showing you the mechanism of binding. Let me just go back and give you more explanation. So this 7 receptor which you have just seen is responsible for transmitting information. And this is a common receptor, and it can take signal from diverse source. Source could be photon. Where do you see photon as a source of signal? Of course, when we are talking about vision, when photon affects our eyes, retina, and then the rhodopsin binding to the 7-TM receptor occurs. Second thing be odorant, that means our smell. Tastant, the one which we taste, the hormones, the neurotransmitter. So you can see that the signal could be of different form. Could be photons, could be odorant, could be tastant, could be hormones, or could be neurotransmitters. So signal could be different. Now, many times these receptors, they will be in the serpentine shape, as you have seen. It goes and traverses through the membrane, and so this is the one which is important for the binding event. And a well characterized member of the family is rhodopsin. Rhodopsin is the one which is important for signal transduction, where photon binding leads to our vision. Now, another example of this is that a receptor that responds to chemical signal is a beta adrenergic receptor. So there is a different form of receptor, but yes, there is also the one where the binding can occur. And this protein binds with epinephrine or adrenaline. You know that many times when sometimes this hormone, which is adrenaline, it is secreted in our body, and we call it as a fight or flight response. What happens? All of a sudden you see something and you respond, and your body has changed its response. So that is what is because of the hormone which is secreted from the body. For example, if you get anger, you listen something, you get angry. During that time, this epinephrine is being secreted and the signal is being transmitted. So thus the binding of these leads to the conformational change in the 7M receptor that can be detected inside the cell. So all these responses, they are mediated through 7TM receptor. Now, looking at the event, yeah, as I said, you have a ligand binds with the receptor. It activates the receptor. Receptor will lead to the activation of G protein. And once G protein is activated, it will activate adenylate cyclase, which will increase cyclic AMP. And what cyclic AMP will do? It will bind and it will increase the activity of protein kinase. And the protein kinase is the one which leads to the modification of enzyme, and that's the way the overall process is happening. So GTP is, in addition to hormone, is essential for signal transduction. As you have seen that GTP is the one which is exchanged with the G protein, and equally revealing was observation that hormone binding stimulates GTP hydrolysis. So what happens? The GTP is the one which is going to be controlling the rate of reaction. It is nothing but it is a G. You know, guanyl. Guanyl is one of the bases which you have learned in nucleic acids. The same is forming GTP and controlling the reaction. The signal coupling protein is termed as a G protein. As I said, G is referred to guanyl nucleotide because it can bind with either GDP or GTP. The activated G protein stimulates the activity of adenylate cyclase, as you have seen here. Once we activate G protein, it can activate adenylate cyclase, and then it increases the concentration of cyclic MP. So from where cyclic MP is formed? Cyclic MP is formed from ATP, which is already present inside the cell. So under inactivated condition or unactivated condition, G nucleotide is bound to G protein in GDP form. This part we have already discussed in the cycle, that initially when G protein is inactive, GDP is bound. In this form, G protein exists as a heterotrimer consisting of alpha, beta, and gamma subunit. The alpha subunit is referred as G alpha. G alpha refers to the alpha bound G protein, which binds to the nucleotide. The role of hormone bound receptors to catalyze exchange of GTP for bound GTP. So what will happen? The inactive G protein, which is binding with the GTP, will be replaced by GTP, and then your gamma and beta subunits, they are being released, and that's the way your activation occurs. The hormone receptor complex interacts with the heteromeric G protein and opens the nucleotide binding site so that GDP can depart, can leave, and GTP can bind from the solution. The dissociation of a G heterotrimer, alpha, beta, and gamma subunit transmits the signal that the receptor has created, and then a single hormone receptor complex can stimulate nucleotide exchange in many G protein heterotrimer. What it means is that here signal is being amplified. One hormone binds with one receptor, and then it can increase the nucleotide exchange in many G protein. Why it is important? That because hormones are required in very, very low concentration, because it can amplify signal. So even in a nanomolar concentration, if hormone is present, they can amplify several G proteins, activation, and this activation will occur. And because of that, we can see that there is a amplification of hormone response. All 7 TM receptors, they are sometimes referred as GPCRs. What is a GPCR? G protein coupled receptor. So the same receptors, because they bind with the G protein, they can also be referred as a GPCRs. So in some books, you will be reading that they are written as GPCRs. So this is the example where this G protein binding to the 7M receptor, what they will do is that this is the structure which is showing that how it can bind with the adenylate cyclase and activate cyclase. So it's a very simple thing. G alpha S. What S refers? S refers that stimulating. In some places you can have G alpha I. G alpha I will refer to inhibitors. So once adenylate cyclase is activated by G protein, they are called as GS, alpha GS. Alpha because there is one subunit which is the one which is in the on form. Now, coming to show you the reaction, what happens? Somewhere here, you have the receptor which is a GPCR. To GPCR, what has happened that your hormone or any other ligand like your photons or, for example, any other signal from the body has bound, and that binding, what it does is that it has activated alpha G protein. So there is a on form of G protein, it binds with adenylate cyclase, and after binding, it can activate the reaction. But what will happen? Once the binding is over, this can be hydrolyzed, and so this G protein has been hydrolyzed to GDP. And once GDP is there, what will happen? This GDP will bind with beta and gamma subunit, and they will be again becoming off. Is the conversion of on and off form. And during this process, what happens? In on form, you can say that adenylate cyclase has opened its active site, and in off form, there is no opening of the active site, and no binding with the G protein. So this reflects how resetting of G alpha occurs by the hydrolysis of GTP. Now your question may be how GTP is being hydrolyzed. The same enzyme, same protein has got enzymatic activity, which very slowly releases a phosphoryl group, and that's the way GTP is getting back to GDP and becomes inactivated. This shows that how the binding etc. occurs. So the dissociation of the as I said, that the binding of the hormone with the receptor will continue the signal. As soon as you remove the hormone from the receptor, when hormone receptor complex is not there, there will be no such activation. Your G protein will not be activated. Another way is that there is an inhibitor which may be present because of the the process, and this is shown that the receptor kinase, they can help in generating beta acertin, and that can lead to the inactivation or termination of the signal. The cyclic AMP is not only second messenger employed, there is another cascade. So far we have talked about G protein, and G protein activation leads to the cyclic AMP activation. Other than cyclic AMP, there is a phosphoinositide cascade. Like adenylate cyclate cascade, there is an extracellular signal which is coming from the phosphoinositide cascade. And this part I have just discussed a few minutes back. Yeah. So in phosphoinositide cascade, what is you have a signal which is IP3 and diacylglycerol. So these are the signals. So binding of the hormone with the receptor, they will secrete these two things. So what they do is that they activate an enzyme which is called the phospholipase, and the phospholipase C will release these two things from the membrane. Then the signal will start. So here it has been shown that the binding of hormones such as a vasopressin, so here hormone is different, binding to the same receptor, it will lead to the activation of beta isoform of phospholipase C. So what phospholipase C will do is that it will cleave the lipid into inositol 1,4,5 triphosphate and diacylglycerol, and each trigger additional step in the signal transduction. So this process is also coupled with the G protein, which brings the catalysis or which activates phospholipase C. There's the whole mechanism given that how protein kinase C. What this C refers to, in case we have discussed earlier, they are called as a protein kinase A. They refer that they are activated by the protein kinase A is activated by adenylate cyclase. On C, this protein kinase is activated by calcium ion. So that is why the term has been referred as a C. And here, when I say protein kinase C, it phosphorylates serine and threonine residue in many target proteins or enzymes. The whole mechanism I'm going to explain, whatever is written here. On PIP2 hydrolysis, the membrane by the enzyme phospholipase C, the domain C1b domain and PKC binds to the DG. So this is very difficult to understand by the writing. I'll explain you how it happens. So what happens that you have a protein kinase C activation. It is a process by which protein kinase C is activated, and once protein kinase C is activated, it will release diacylglycerol and IP3 from the membrane. How it happens? You have a diacylglycerol binding site which has a zinc ion. Then you have inactive protein kinase C in a solution. Now, inactive protein kinase C, the active site of protein kinase C is bound with the pseudo substrate, which is a part of the the system. And what happens as soon as this protein kinase C can bind with the membrane? Where do they bind? They bind to the diacylglycerol. So these are the component where diacylglycerol is there. So it has been shown that this is a diacylglycerol binding site, And then what happens, the protein kinase C, the third domain. So these are different domains, 1, 2, 3, 4 domains, where two of them, they are binding with the diacylglycerol, one of them binds with the calcium ion, and third one is the one which is responsible for activation of the reaction. Now, calcium is an important component of one signal transduction circuit, many cases in phosphoinositide cascade. What it means is that even inside the cell, calcium helps in the activation of protein kinase C. And this the cytosolic level of a unexcited cell is around 100 nanomolar. Now, when they are excited, they can become much, much higher, several folds higher. For example, in blood, its magnitude is a 5 millimolar. So you can see the difference in the concentration. And this steep concentration gradient present in the cell with gives a lot of opportunity. And the cytosolic concentration of calcium ion can be abruptly raised from signaling process. And how it can happen? Because in the blood you have such a high concentration compared to the unexcited cell, what you can do is that you can open the calcium channel. You have learned about the ion channel. As soon as signal comes, calcium ion channel can open and the calcium ion concentration can increase inside the cell. So calcium ion is the one which is a regulator of enzyme like protein kinase C. This is the one which shows there is another protein which can also be regulatory. This protein is known as a calmodulin protein, which is a calcium dependent protein. And that's why it is important that not only protein kinase C, calmodulin can also be responsible, which can stimulate many enzymes and transporters. So here you can see that this is a CAM target peptide, which is present in enzyme, which is called as a calmodulin kinase 1, and this is calmodulin. As soon as it is inactive form, when calcium binds, it becomes in a proper structure which can actually bind with the CAM kinase. So it is showing that how CAM kinase is bound to this. So you can see, I will just try to show it to you. So this is the enzyme which gets activated after calcium binding. This is the calmodulin, which is a calcium binding protein. So once this calcium binding protein is bound to calcium, becomes activated and can bind with a CAM kinase peptide, and that's the way the whole reaction starts. So this way we have covered how the signal transduction occurs. To sum up, you have learned that hormone receptor binding can lead to the activation of G protein. G protein in turn can be regulated itself, which can be either on form or off form. In the on form, it is bound with the GTP, and it can come to the off form in two stages. One, either hormone receptor has been released, the receptor is not bound with the hormone, that can terminate the signal. Another way could be that GTP is a slowly, so when hormone is not binding to the receptor, that means more activation of G protein will not occur. On the other hand, G protein, which is already activated, will be releasing its GTP and it will be becoming GDP, and thereby it will be becoming inactivated. So that's the one way. Now, once G protein is activated, you have learned either it can go through the adenylate cyclase pathway, where it can activate protein kinase, and then further it can activate, the other increase the cyclic MP and then can increase. Alternatively, the activation of G protein through other hormone or other ligand can also lead the pathway through the cascade, which releases diacylglycerol or calcium ion or IP3. So all these three things are important as a signal transducing mechanism. You have learned that the phosphatidyl inositol pathway or inositide cascade is responsible for the mechanism of adrenaline, where adrenaline bind with the hormone receptor and can activate the signal, and this pathway happens. Now, as I said, that this pathway is cascade. That means one leads to another, to another, to another, and every time signal is being amplified. That means if I have 1 nanomolar here, the response could be 10 nanomolar here, 100 nanomolar here, and 1000 nanomolar here. And so it will be in micro, nano, micro, and the millimolar concentration. So the response will be equal to very high concentration of hormone. So with this, I'll stop at this point. If you have any question, we can discuss.
Sir, I had questions in protein purification.
Yeah, बताइए.
Yes, sir.
So what is the question?
Sir, in electrophoresis, sir, there is the mention of that the gel acts as a molecular sieve. So, and that so that proteins move according to charge to mass ratio. I was not able to understand that.
See, what I mean to say here is that if I take any protein, each protein will have some charge, isn't it?
Yes, sir.
Because they will differ in terms of the amino acid they have. So based on the amino acid, they will be having certain charge. But later on, what happens, if I want to separate the protein based on the charge, the electrophoresis is not the method. What happens, all the charge is present in the protein, they are neutralized. How? They are binding to SDS, sodium dodecyl sulfate or sodium lauryl sulfate. Now here, all the protein will have the same charge, or at least you can say that the charge on the surface of protein will be neutralized, and that will be minimum. Their effect will be minimum, and more will be on the basis of sodium dodecyl. So now each protein, depending on the molecular mass, they will be having a charge. Why? Because I said that one amino acid can bind with, say, 1.4 SDS and molar concentration. So what will happen? More the number, more the charges. So based on the size of the protein, they will be separated in electrophoresis.
Okay,
Okay.
sir. Yes, sir. Sir, and
Yeah.
what are ampholites? It was mentioned in isoelectric focusing.
Yeah. See, ampholites are the one which has got both positive charge and negative charge. So these are the the polymers. It's basically a polymer which can be present on the surface of the separating electrophoretic unit. And what will happen? This will create a gradient of pH. So what will happen from one to another ampholyte, that is a solution, and this will create a gradient of pH, and pH gradient will be as low as 0.1 pH. That means even if there is a difference of a single charge in the protein, they can be separated using isoelectric focusing.
Okay, sir. Sir, one more question, sir. Sir, in the metabolism part of the syllabus, sir, that is from introduction to metabolism up till last class. So, sir,
Yeah.
what kind of questions will you ask? Will you, matlab,
See, now say it is an open book, so there is no problem. Whatever I'm asking you to understand the concept, and whatever is being taught, you should be able to, give answer to that. The
oh,
metabolism, you may not have a direct question, or because your concept should be clear. That is most important, and that's why you may have seen in your quizzes, the question asked is based on your understanding.
yes,
And if you understand, then only you can answer. You cannot just copy an answer. I have seen many of you, or I'm not talking about you alone, I'm talking about general in a class. People, they just go and try to search through Google. You get absolutely incorrect answer. So I would say that it is important that you should read the book, you should read the class notes, because what I have taught you, 99% I have taught you from the Destroyer's book, and some percentage I have taken from Leninger's book. Both the books are level. That means both of them are excellent book in the biochemistry. You refer, you clear your concept so that you can answer the queries. Any more
sir.
question?
Yes, sir, sir. So, sir, pentose phosphate pathway would be activated during cell division for nucleic
If,
acid.
see, whenever the cell division occurs, there are as I said, there are different types of pentose phosphate pathway. They are, coupled with your glycolysis. So there are different mechanism which occurs. When suppose the nucleic acid synthesis occurs and you require more of, so it is not actually we are looking at nucleic acid synthesis. What we are looking at is that whether we need both ribose unit as well as the NADPH simultaneously in the equivalent amount, or we are looking at just ribose units, or we are looking at just NADPH. So based on that, there is an interlink between the different pathway, which is a glycolysis and pentose phosphate pathway.
Okay, sir. Thank you, sir. So that is all the questions I have.
Anyone else?
No, sir.
So you are still open to the question. You can write the question. Whatever the question you have, you can even ask after the quiz in the evening. So that is open. You should, in fact, once you read all this, you will get several questions in your mind. And whatever the questions you have, you are most welcome to ask, and I will be happy to answer you.
What will be the timing for the major?
Major, as per your timetable, it is from 10 to 12. So 12 is the time for submission. I will give you another 15 minutes before the start of the examination.
So, sir, 10 to 12, 15. Okay, sir.
Not 10 to 12, it is 9:45 to 12.
Okay, sir.
So that all the message you will get in advance.
Okay, sir.
And I request everybody to submit only in Microsoft Teams, because otherwise it creates a lot of problem in the sense that management. I do not know time. If I don't know the correct time, it will not be evaluated, number one. Number two, it is not justified for others who are submitting in time, because anybody, if I give you 1 hour extra, everybody can solve the question paper, because it is unlike solving a mathematical question. It is just thinking about the concept, and quick concept revival can be done if you have gone through your course. So if you study, there is no problem. You can answer all the question in a time. I calculate myself that if I have to write, if I know answer, and even if I have to think a little bit, how much time I will take for per question. And based on that, questions are being framed. They are of different levels. Some of the question I know all of you have solved, but some of the question only 10% or 20% of you can solve. So that's the way the questions will be at different level. And yes, if you study, I think you can do excellent. So if there are no more questions, thank you very much. And then I look forward for.
Thank you, sir.
Thank you, sir.
Thank you. Bye.